Ebola, also known as Ebola virus disease (EVD), or Ebola hemorrhagic fever (EHF), is a viral hemorrhagic fever of humans and other primates caused by Ebola viruses. Signs and symptoms typically start between 2 days and 3 weeks after contracting the virus, with a fever, sore throat, muscular pain, and headaches. Vomiting, diarrhea and rash usually follow, along with decreased function of the liver and kidneys. At this time, some people begin to bleed both internally and externally. The disease has a high risk of death, killing 25% to 90% of those infected, with an average of about 50%. This is often due to shock from fluid loss, and typically follows 6 to 16 days after symptoms appear. Quick facts, other names, specialty. The virus spreads through direct contact with body fluids, such as blood from infected humans or other animals. Spread may also occur from contact with items recently contaminated with bodily fluids. Spread of the disease through the air between primates, including humans, has not been documented in either laboratory or natural conditions. Semen or breast milk of a person after recovery from EVD may carry the virus for several weeks to months. Fruit bats are believed to be the normal carrier in nature, able to spread the virus without being affected by it. Other diseases such as malaria, cholera, typhoid fever, meningitis and other viral hemorrhagic fevers may resemble EVD. Blood samples are tested for viral RNA, viral antibodies or for the virus itself to confirm the diagnosis. Control of outbreaks requires coordinated medical services and community engagement. This includes rapid detection, contact tracing of those who have been exposed, quick access to laboratory services, care for those infected, and proper disposal of the dead through cremation or burial. Samples of body fluids and tissues from people with the disease should be handled with special caution. Prevention includes limiting the spread of disease from infected animals to humans by handling potentially infected bushmeat only while wearing protective clothing, and by thoroughly cooking bushmeat before eating it. It also includes wearing proper protective clothing and washing hands when around a person with the disease. An Ebola vaccine was approved in the United States in December 2019. While there is no approved treatment for Ebola as of 2019, 2 treatments, atoltuvimab, martuvimab, odosivimab and ansuvimab, are associated with improved outcomes. Supportive efforts also improve outcomes. This includes either oral rehydration therapy, drinking slightly sweetened and salty water, or giving intravenous fluids as well as treating symptoms. Atoltuvimab, martuvimab, odosivimab, Inmazeb, was approved for medical use in the United States in October 2020, for the treatment of infection caused by Zaire Ebola virus. The disease was first identified in 1976, in 2 simultaneous outbreaks, one in Nzara, a town in South Sudan, and the other in Yambuku, Democratic Republic of the Congo, a village near the Ebola River from which the disease takes its name. EVD outbreaks occur intermittently in tropical regions of sub-Saharan Africa. From 1976 to 2012, the World Health Organization reports 24 outbreaks involving 2,387 cases with 1,590 deaths. The largest outbreak to date was the epidemic in West Africa, which occurred from December 2013 to January 2016, with 28,646 cases and 11,323 deaths. It was declared no longer an emergency on the 29th of March 2016. Other outbreaks in Africa began in the Democratic Republic of the Congo in May 2017 and 2018. In July 2019, the World Health Organization declared the Congo Ebola outbreak a World Health Emergency. Signs and symptoms. Onset. The length of time between exposure to the virus and the development of symptoms, incubation period, is between 2 and 21 days, and usually between 4 and 10 days. However, recent estimates based on mathematical models predict that around 5% of cases may take longer than 21 days. Ebola viruses contain single-stranded, non-infectious RNA genomes. Ebola virus genomes contain 7 genes including 3, utrnpvp3 5vp4 ogpvp3 ovp24l5, UTR. The genomes of the 5 different Ebola viruses BDBV, EBOV, RESTV, SUDV and TAFV differ in sequence and the number and location of gene overlaps. As with all filoviruses, Ebola virus virions are filamentous particles that may appear in the shape of a shepherd's crook, of a, U, or of a, 6, and they may be coiled, toroid or branched. In general, Ebola virions are 80 nanometer, in width and may be as long as 14,000 nanometers. Their life cycle is thought to begin with a virion attaching to specific cell surface receptors such as C-type lectins, DC sign, or integrins, which is followed by fusion of the viral envelope with cellular membranes. The virions taken up by the cell then travel to acidic endosomes and lysosomes where the viral envelope glycoprotein GP is cleaved. This processing appears to allow the virus to bind to cellular proteins, enabling it to fuse with internal cellular membranes and release the viral nucleocapsid. The Ebola virus structural glycoprotein, known as GP1, 2, is responsible for the virus's ability to bind to and infect targeted cells. The viral RNA polymerase, encoded by the L gene, partially uncoats the nucleocapsid and transcribes the genes into positive strand mRNAs, which are then translated into structural and non-structural proteins. The most abundant protein produced is the nucleoprotein, whose concentration in the host cell determines when L switches from gene transcription to genome replication. Replication of the viral genome results in full-length, positive strand antigenomes that are, in turn, transcribed into genome copies of negative strand virus progeny. Newly synthesized structural proteins and genomes self-assemble and accumulate near the inside of the cell membrane. Virions bud off from the cell, gaining their envelopes from the cellular membrane from which they bud. The mature progeny particles then infect other cells to repeat the cycle. The genetics of the Ebola virus are difficult to study because of EBOV's virulent characteristics. Transmission. It is believed that between people, Ebola disease spreads only by direct contact with the blood or other body fluids of a person who has developed symptoms of the disease. Body fluids that may contain Ebola viruses include saliva, mucus, vomit, feces, sweat, tears, breast milk, urine and semen. The WHO states that only people who are very sick are able to spread Ebola disease in saliva, and the virus has not been reported to be transmitted through sweat. Most people spread the virus through blood, feces and vomit. Entry points for the virus include the nose, mouth, eyes, open wounds, cuts and abrasions. Ebola may be spread through large droplets, however, this is believed to occur only when a person is very sick. This contamination can happen if a person is splashed with droplets. Contact with surfaces or objects contaminated by the virus, particularly needles and syringes, may also transmit the infection. The virus is able to survive on objects for a few hours in a dried state, and can survive for a few days within body fluids outside of a person. The Ebola virus may be able to persist for more than 3 months in the semen after recovery, which could lead to infections via sexual intercourse. Virus persistence in semen for over a year has been recorded in a national screening program. Ebola may also occur in the breast milk of women after recovery, and it is not known when it is safe to breastfeed again. The virus was also found in the eye of one patient in 2014, 2 months after it was cleared from his blood. Otherwise, people who have recovered are not infectious. The potential for widespread infections in countries with medical systems capable of observing correct medical isolation procedures is considered low. Usually when someone has symptoms of the disease, they are unable to. Vaccines. An Ebola vaccine, RVSV-ZEBOV, was approved in the United States in December 2019. It appears to be fully effective 10 days after being given. It was studied in Guinea between 2014 and 2016. More than 100,000 people have been vaccinated against Ebola as of 2019. Infection control. Caregiver. People who care for those infected with Ebola should wear protective clothing including masks, gloves, gowns and goggles. The U.S. Centers for Disease Control, CDC, recommend that the protective gear leaves no skin exposed. These measures are also recommended for those who may handle objects contaminated by an infected person's body fluids. In 2014, the CDC began recommending that medical personnel receive training on the proper suit-up and removal of personal protective equipment, PPE. In addition, a designated person, appropriately trained in biosafety, should be watching each step of these procedures to ensure they are done correctly. In Sierra Leone, the typical training period for the use of such safety equipment lasts approximately 12 days. Patients and household members. The infected person should be in barrier isolation from other people. All equipment, medical waste, patient waste, and surfaces that may have come into contact with body fluids need to be disinfected. During the 2014 outbreak, kits were put together to help families treat Ebola disease in their homes, which included protective clothing as well as chlorine powder and other cleaning supplies. Education of caregivers in these techniques, and providing such barrier separation supplies has been a priority of Doctors Without Borders. Disinfection. Ebola viruses can be eliminated with heating for 30 to 60 minutes at 60 degrees Celsius or boiling for 5 minutes. To disinfect surfaces, some lipid solvents such as some alcohol-based products, detergents, sodium hypochlorite, bleach, or calcium hypochlorite, bleaching powder, and other suitable disinfectants may be used at appropriate concentrations. General population. Education of the general public about the risk factors for Ebola infection and of the protective measures individuals may take to prevent infection is recommended by the World Health Organization. These measures include avoiding direct contact with infected people and regular hand washing using soap and water. Bushmeat. Bushmeat, an important source of protein in the diet of some Africans, should be handled and prepared with appropriate protective clothing and thoroughly cooked before consumption. Some research suggests that an outbreak of Ebola disease in the wild animals used for consumption may result in a corresponding human outbreak. Since 2003, such animal outbreaks have been monitored to predict and prevent Ebola outbreaks in humans. Corpses burial. If a person with Ebola disease dies, direct contact with the body should be avoided. Certain burial rituals, which may have included making various direct contacts with a dead body, require reformulation so that they consistently maintain a proper protective barrier between the dead body and the living. Social anthropologists may help find alternatives to traditional rules for burials. Transport, travel, contact. Transportation crews are instructed to follow a certain isolation procedure, should anyone exhibit symptoms resembling EVD. As of August 2014, the WHO does not consider travel bans to be useful in decreasing spread of the disease. In October 2014, the CDC defined 4 risk levels used to determine the level of 21-day monitoring for symptoms and restrictions on public activities. In the United States, the CDC recommends that restrictions on public activity, including travel restrictions, are not required for the following defined risk levels. Having been in a country with widespread Ebola disease transmission and having no known exposure, low risk, or having been in that country more than 21 days ago, no risk. Encounter with a person showing symptoms but not within 3 feet.